Eukaryotes includes organisms like protists, plants, animals and fungi. Plasma membrane contains what? 52% of protein and 40% of lipids. So one cell communicates with the neighboring cell with the help of Plasmodesmata, hence they are called as cytoplasmic channels. Hello dear students, welcome back to session 1 of this chapter called Cell, the Unit of Life. Myself, B. Shobharani from the Department of Biology, Vidhyaashram Pre-University College, Mysuru, the Temple of Excellence. What are we going to study in this today's session? I think in the last session, I have discussed about the prokaryotic cell and a eukaryotic cell. So in today's session, moving along with the remaining concepts of this chapter called the cell, the unit of life. Now let us understand what is the difference once again between a prokaryotic cell and a eukaryotic cell. A typical true nuclear membrane is absent. Here a true nuclear membrane is always present. So what is the first very important difference between a prokaryotic cell and a eukaryotic cell? A typical true nuclear membrane is absent in prokaryotic cell, but it is present in eukaryotic cell. Membrane bound organelles such as mitochondria, plastids, ER, Golgi bodies, etc. are absent. As I said, all the cell organelles are absent except for the ribosomes. So here generally all membrane bound organelles are present. Here it is absent, there it is present. Next, ribosomes are of 30, as I said, ribosomes are present both in a prokaryotic cell and a eukaryotic cell. So the ribosomes are of 30S and 70S types. Here the ribosomes are of 40S, 60S and 80S types. And cell division is of simple binary fission. Here cell division is complex with involvement of chromosomes. Here, what do you mean by cell division of simple binary? Like for example, you take bacteria. So it is one cell divides into two. So it is a very simple type of division. Here cell division is complex with involvement of chromosomes. So important examples are what? Bacteria, cyanobacteria and mycoplasma. Here what are the examples you are going to give? Includes all groups of higher plants and animals. So I repeat, what is so very important differences between a prokaryotic cell and a eukaryotic? This is a very important concept from examination point of view. So a typical and true nuclear membrane is absent there. A true nuclear membrane is always present. Membrane bound organelles such as mitochondria, plastids, endoplasmic reticulum, Golgi bodies, etc. are absent. There generally all membrane bound organelles are present. Ribosomes are of 30S, 70S types. Their ribosomes are of 40S, 60S and 80S types. Here cell division is of simple binary fission. There cell division is complex with involvement of chromosomes. Important examples are what? Bacteria, cyanobacteria and mycoplasma. There includes all groups of higher plants and animals. Now moving on to the eukaryotic. So here you can see the beautiful picture of a eukaryotic cell. Both a plant cell and an animal. So this is a picture of a plant cell. Here you can see the cell wall. This is a plant cell. See the beautiful picture of a plant cell. What are all the parts that are found in a plant cell? Here you can see the nucleus, nucleolus, Golgi bodies, nuclear envelope. See here you can see the nuclear envelope. Hence they are called as what? Eukaryotic cell. Then you can see the vacuole. So vacuole is present in plant cell but absent in animal cell. So middle lamellae, cell wall, mitochondria, ribosomes, chloroplasts. Then we have perioxysome which help in respiration, microtubules, plasmodesmata. Plasmodesmata act as cytoplasmic connections. So they communicate with each other through cytoplasmic connections. Then smooth endoplasmic reticulum, rough endoplasmic, as I said, here you can see the difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. On rough endoplasmic reticulum, you can come across the ribosome. Ribosomes are present, hence the name rough endoplasmic reticulum. In case of smooth endoplasmic reticulum, the ribosomes are absent. So moving across the eukaryotic cell. A cell with a defined nucleus is called eukaryotic cell, and eukaryotes includes all what? Protists, plants, animals and fungi. Extensive compartmentalization due to membrane-bound cell organelles possesses an organized nucleus with nuclear envelope, as I said. So I repeat, a eukaryotic cell is a cell with defined nucleus. Eukaryotes includes organisms like protists, plants, animals, and fungi. Then extensive compartmentalization due to membrane-bound cell organelles possess an organized nucleus with nuclear envelope. Here you can see the structure of an animal cell. So that was about, the previous diagram was about the plant cell, and this is an animal cell. This is an animal cell. So here you can see, in plant cell, the outermost layer was it is covered over by cell wall. Here it is covered by plasma membrane. Here you can see centriole, perioxysome, but you don't find chloroplast and you don't find vacuole. Right? So we have these ribosomes, mitochondria, rough endoplasmic reticulum, cytoplasm, nucleolus. See, as you know, it is a eukaryotic cell. It has a nuclear membrane. It has mitochondria, smooth endoplasmic reticulum, then rough endoplasmic reticulum. So have a variety of complex locomotory and cytoskeletal structures. So genetic materials, as you know, they are organized in the form of chromosomes, where we use the word called chromatin, right? So the plant cell possesses cell wall, plastids, and a large central vacuole, which are absent in plant cell. So what is a very important difference between a plant cell and an animal cell? Cell wall is present in plant cell wall is absent in animal cell. Then vacuoles are present in plant cell, absent in animal cell. Then chloroplast is found in plant cell because they manufacture their own food. But eukaryotic cells are heterotrophic in nature, so they don't have the chloroplast. So animal cells have centrioles, which are absent in plant cells. Then cell membrane, a very, very important, as I said, the cell wall, the cell membrane. The cell membrane is composed of lipids that are arranged in bilayer. Here you can see the lipid layer, 2 layers. So it is called bilipid layer, right? And lipids are arranged within the membrane with the hydrophilic polar ends towards the outer sides and the hydrophobic tails towards the inner part. So here you can see the hydrophilic polar tails towards the outer sides, right? Then and the hydrophobic tails towards the inner part, right? Here you can see hydrophobic. Yes, here you can see the peripheral protein, right? Then non-polar tail of saturated hydrocarbons is protected from the aqueous environment. This is a very important diagram from examination point of view. So here the lipid component of the membrane mainly consists of what? Phosphoglycerides. It is called phosphoglycerides. So the cell membrane also possesses protein and carbohydrates. Ratio of protein and lipids varies from cell to cell. So the cell membrane is composed of lipids that are arranged in bilipid layer. Then lipids are arranged within the membrane with the hydrophilic polar tails towards the outer side, and towards the inner side you can see the hydrophobic tail towards the inner part, and non-polar tail of saturated. Here you can see the non-polar saturated hydrocarbons protected from the aqueous environment. And the lipid component of the membrane mainly consists of what? Phosphoglycerides. Cell membrane also possesses protein and carbohydrates. Ratio of protein and lipids varies from cell to cell. Moving across the remaining part, so human erythrocyte plasma membrane contains 52% of protein and 40% of lipids. So when we come across human erythrocytes, plasma membrane contains what? 52% of protein and 40% of lipids. So here membrane protein may be integral proteins or partial or totally buried in the membrane. Here you can see the integral proteins. Here you can see the integral proteins. They are either partially or totally buried in the membrane. Here you can see these both are the integral proteins. And these are the bilipid layers. Bilipid layer. This is the bilipid. This is the bilipid layer. Here you can see the peripheral proteins, hydrophobic ends, you can see, right? then the improved model of the surface and integral proteins are partially or totally buried in the membrane. So the improved model of structure of this plasma membrane was proposed by Singer and Nicholson, two scientists by name Singer and Nicholson in 1972, widely accepted as, this is called as fluid mosaic model. This is called as fluid mosaic model. And according to this, the fluid nature of lipid enables the lateral movement of proteins within the overall bilayer. And this model, as I said, it was proposed by Singer and Nicholson in 1972. So what is this model all about? According to this model, mosaic of proteins are embedded in the fluid bilayer of phospholipids. Here you can see the integral proteins. They are like mosaic, hence the name fluid mosaic model. And the polar hydrophilic heads of phospholipid molecules face the free surface and the non-polar hydrophobic tails face the inner surface. And in between two lipid layers, number of proteins, and some are embedded in the lipid layer called intrinsic proteins, and some are apart from the lipid layer called extrinsic proteins. So here you can see the intrinsic proteins and the extrinsic proteins, right? So in some glycoproteins and glycolipids bound to the membrane surface as cell identity markers. So I repeat this model, the fluid mosaic model, which is very important from examination point of view. Definitely they are going to ask this particular concept for examination. So this model was proposed by Singer and Nicholson in 1972, and according to this model, mosaic of proteins are embedded in the fluid bilayer of phospholipids, and the polar hydrophilic heads of phospholipid molecules face the free surface and the non-polar hydrophobic tails face the inner side, and in between two lipid layers, number of proteins, and some are embedded in the lipid layer called intrinsic proteins, and some are apart from the lipid layer called extrinsic proteins. So some glycoproteins and glycolipids bound to the membrane surface as cell identity markers. So what is the function of this cell membrane then? As we know, it is very, very important. It helps in passive transport, that is, many molecules can move briefly across the membrane without any requirement of energy. So hence it is called as what? Passive transport. What do you mean by active transport and passive transport? If at all, if the process requires energy in the form of ATP, it is called active transport. If it doesn't require energy in the form of ATP, it is called passive transport. So cell membrane is acting as a passive transport where many of the molecules move briefly across the membrane without any requirement of energy. So like it takes place through simple diffusion along the concentration gradient. Like what is diffusion? It is a movement of molecules from a region of higher concentration to a region of lower concentration. And also it helps in facilitated diffusion. What do you mean by facilitated diffusion? There are some protein molecules with which act as channels. So with the help of protein channels, the process of diffusion takes place, and such type of diffusion is called facilitated diffusion. And sometimes through active transport, like a few ions or molecules are transported across the membrane by the expenditure of lower concentration through semi-permeable membrane. Right. Next, cell wall. Now let us understand about cell wall. So that was about cell membrane. So what is cell wall? It is a non-living rigid structure called cell wall present outside the plasma membrane of the plant and a fungal cell. Algae have a cell wall made up of cellulose, galactans, mannan, and the minerals like calcium carbonate. So plant cell wall consists of what? Cellulose, hemicellulose, pectin, and protein. So this is the chemical composition of the plant cell wall. As you know, the cell wall of the young plant is called primary cell wall. On maturity, secondary cell wall formed inner to it. As you know, compared to the animal cell, the plant cell wall is more rigid, and that rigidity is due to the presence of the chemical substance called cellulose and hemicellulose added by additional deposition of pectin. So it is called pectinization. They all form the primary cell wall material, that is cellulose and hemicellulose. If there is an additional deposition of a chemical substance called pectin, then we call it as pectinization. Sometimes even lignin, for example, like in case of sclerenchyma, which gives mechanical support, then it is deposited by lignin. So the middle lamella, so in between this we have this middle lamella. So this middle lamella is a layer of calcium pectate, which holds or glues the neighboring cells. The cell wall and middle lamellae may be traversed by plasmodesmata. Very, very important. Plasmodesmata, they act as cytoplasmic channels or connections, right, between the two adjacent cells. So plasmodesmata, as I said, they are microscopic narrow channels found in the cell wall of certain plant cells that act as intracellular. So one cell communicates with the neighboring cell with the help of plasmodesmata, hence they are called as cytoplasmic channels. So one cell will be communicating with the neighboring cell with the help of this plasmodesmata, which bridges or facilitate communication and transport of materials between the plant cells. Hope you have understood the concepts what I have explained in today's session, particularly about the fluid mosaic model put across by Singer and Nicholson. So in the coming session, I'll be dealing with the remaining concepts of this very interesting chapter called Cell, the Unit of Life. Till then, goodbye and thank you.